This video will demonstrate an example of how to analyze variants using the gene portals. In this video, we will specifically show how to navigate the GRIN portal. However, the same techniques can be applied to the other gene portals as they operate very similarly. The first example will demonstrate the analysis of a protein truncating variant, and the second example will demonstrate the analysis of a missense variant. To begin, select your gene of interest and click the green search button, which will take you to the portal of interest. Select the Variant Analysis tab at the top of the page in order to begin analyzing your desired variant. Select your gene of interest and the corresponding transcript using the drop-down boxes on the left side of the page. Then, enter your variant on either the cDNA or protein level and click the green search button to search for your variant. On the right side of the page, a variant summary will appear showing the gene, detailed effect, ACMG classification, inheritance, and phenotype specificity. You have the ability to modify the inheritance or phenotype specificity based on the information you have available, which in turn refines the ACMG classification. For example, if you know the variant occurred de novo, select the de novo button. Notice that this alters the ACMG classification in this example from likely pathogenic to pathogenic. These modifications also are reflected in the Customize ACMG Criteria and Explore Data tab. In addition to modifying the inheritance and phenotype specificity on the Variant Summary tab, the Customize ACMG Criteria and Explore Data tab will also allow you to modify criteria based on available information. For example, if you know that PM1 should be applied, you can click Edit in order to edit the strength. Notice that the PM1 box is now showing in red, indicating that this criteria is now applied. Next, clicking through the Cohort Information tab allows you to view other individuals with the same variant. By clicking on the Compare Phenotypes tab, you can explore what phenotypes have been associated with patients in our cohort that have a similar variant. Further phenotypic information can also be found under the Basic Information tab at the very top of the page.
This tutorial will demonstrate how the gene portals work for a missense variant. Given the available information on the variant selected to view in this portal, we will explore how to implement this into the portal. First, select the gene of interest. Once you select your gene of interest, the green button will guide you to the GRIN portal of choice. Now we will explore this portal to exemplify the use of variant analysis. In the Variant Analysis tab, the gene and transcript is pre-populated for GRIN2B. If needed, select your gene of interest and the corresponding transcript using the drop-down boxes on the left side of the page. Enter either the variant cDNA level or the variant protein level based on the available information given. Be sure to select the inheritance type, which is de novo, maternal, and paternal confirmed. Once the given variant information is selected and entered, we will now explore what information the variant analysis can tell you. Under the variant summary, the ACMG classification can be obtained, which is currently preliminary. Some information regarding the variant itself includes specifically the clinical significance. Under the cohort information, variants collected here will show individuals with the same variant in this portal, functional information, and individuals with pathogenic variants in the Paragulus GRIN genes available to view in this tab. Similar to the previous example, you can modify inheritance and phenotype specificity, which will refine the ACMG classification. Additionally, you can customize the ACMG criteria and Explore Data tab, which will also modify the ACMG classification. In this tab, it includes the user-defined criteria supporting the pathogenic evidence PP2 and strong PS2 inheritance evidence. It also includes the population data, the patient data, along with computational predictions, including the Revel scores, supporting the PP3 evidence of the chosen variant compared to the control and pathogenic patient cohort. It also shows mutational hotspots and functional data predicting the GRIN function selected variant with increased and decreased function. Under the Comparative Information tab, the comparison of the selected variants with other similar variants in the GRIN2B genes will include information framework enabling clinical and molecular readout comparisons associated with variants affecting the same residue, same domain, same functional consequence, or the same variant type.